In the last couple lessons, I've given you a lot of information about what we already know pretty confidently about neurodegenerative diseases. But I want to back it up and review a bit about how we as a field have reached these conclusions. This is important because we have spent decades upon decades piecing together information across studies and model systems, and I want you all to think about what the future holds for neurodegenerative research. So today we're going to walk through the ways we've gotten to where we are. The field, of course, started by observing patients presenting with these diseases. Since then, we have distinguished sporadic cases, or cases that have no known cause, versus familial cases, ones that we know are inherited. We've learned a lot about the cellular mechanisms by using in vitro experiments or looking at cells in a dish. And a huge portion of what we know comes from in vivo experiments using living model systems, including Drosophila, which are fruit flies, rodents, and non-human primates. Lastly, in our new age of technology, computational and predictive models are becoming more and more common. I won't be talking about those today for the sake of time. We're going to start by jumping way back in time over 100 years ago to the first described cases. So Dr. Alois Alzheimer described a patient he had in 1906 by the name of Mrs. Augustus Deter. In this case study, which is a study of one person, he described her symptoms as they progressed over time, specifically highlighting a rapidly worsening memory, being disoriented to time and place, often confused, and he noted that her state worsened over time until she was completely unintelligible and eventually totally apathetic. And when she died, Dr. Alzheimer was able to look at her brain, where he observed the hallmark plaques and tangles. Of course, he didn't know at the time what exactly these were, but he did see them. And these are actually his drawings that he made depicting what he saw in the brain tissue. Other neurodegenerative diseases have similar origin stories. Dr. James Parkinson actually described Parkinson's all the way back in 1817, calling it the shaking palsy because of tremors he observed in his patients. It wasn't until 1912, though, that Dr. Louis looked at the brains of patients with the shaking palsy and described the aggregates. By now, I'm sure you're starting to see a pattern with how these diseases and their components were named. So I'm sure you can guess who first described Huntington's disease. So in 1872, Dr. George Huntington was on a trip to Long Island, New York, and he saw women from his buggy literally dancing down the side of the road. Asked the person who he was with why they were dancing in such an odd way. The driver told him about this family that lived out on Long Island that had these uncontrollable movements, and Dr. Huntington investigated, eventually publishing his paper on chorea. Now, we've come a long way since these diseases were first observed and described in the wild. I mentioned before that some major discoveries were made because researchers discovered families in which these diseases were almost certain to appear, which led to experimenters and scientists studying these families to identify the possibility of genetic or inherited causes, which we now refer to as familial cases. This is a very famous family tree here, coming from a paper in 1997, which led to the identification of a mutation in the SNCA gene, the gene that codes for alpha synuclein, causing Parkinson's disease. So in this family tree, the people indicated with filled in circles and squares are family members that developed the disease, whereas these clear ones did not. Down here, we see a list of family members identified by number. And if we look at the genetic test results below, the numbers corresponding directly below, we see that all the people who developed Parkinson's disease have these extra bands down here, indicating this genetic mutation in the SNCA gene. This was huge for giving researchers an idea of what they should be looking for in patients and where they should be looking, and is the reason why we now focus so much on alpha synuclein. Familial cases can only get us so far, though. It's clear that inherited cases actually account for very few of the total number of cases, which means we need to think wider than just within families. This is where genome-wide association studies, or GWAS studies, come in. These experiments are intended to cast a wide net. I'm talking thousands to tens of thousands of individuals. This requires a population of people that do not present with the disease and a population that does. And by genotyping these two groups, researchers can identify if any genes show up as different. This is talked about by saying we find genes that are enriched or occur at a higher rate, or reduced, occurring at a lower rate in the diseased population compared to a quote unquote normal population. This chart here shows the results of many GWAS studies, supplemented by other types of studies, that have identified genes related to Alzheimer's disease. Up in this corner, you see mutations that 100% cause Alzheimer's disease, and you'll recognize APP from our last lesson. However, there are many genes that don't 100% cause the disease, but rather increase a person's risk of Alzheimer's disease, which are all of these down here. On the x-axis, we see the rarity of the gene, or how common it is seen in the public. This has led to the identification of many of these genes, from rare to common, at their risk low to high, that lead to Alzheimer's disease. For example, ApoE4. ApoE4 is now highly studied and to try to determine why it is a bigger risk to develop Alzheimer's disease compared to other forms of the ApoE gene, ApoE3 and ApoE2. One thing I do want to note here is that many GWAS studies, or any study using humans that has led to the development of charts like this, have been greatly skewed to focus on white populations. So ApoE4 is actually only a common large risk factor to develop Alzheimer's disease in white and Asian populations. This does not hold true for Black or Hispanic populations, which is something that is important to note about how bias exists in research, health, and medicine. Now, there are many more types of studies than just genetic studies in human populations. It's entirely possible to build a study, say, comparing rates of Parkinson's disease between vegetarians and omnivores, or people who only eat organic vegetables and those who do not pay special attention to organic or not. But we need to think hard about the benefits and drawbacks of deciding to use human populations to investigate our hypotheses. We'll talk about more during our synchronous class time, but a pro that immediately comes to mind is accuracy. These are diseases that affect people, so studying people is studying the disease. On the flip side, you give up a lot of control over the population. You essentially have to take people at their word that they eat only organic food, for example. And if you try to control it a bit by giving people organic food, maybe, you still run into possible problems where they may only kind of follow your directions. You know, maybe they grab an apple to snack on at work because they didn't pack a lunch. There's no way for you to force a person to follow your directions 100%. And one more negative that comes to mind is time. Studying people takes a long time, especially when we're talking about diseases in aged population that take years to decades to progress. This means that people have had to find a way to study these diseases at a more controllable, faster timescale. Starting small, I want to talk about in vitro experiments, which literally mean in glass experiments. You may have also heard of cell culture experiments. So these are experiments where people grow cells in a dish to study their hypotheses. For example, we can grow neurons in a dish and observe what they look like over time. We can also manipulate them in different ways, such as pouring toxic chemicals or compounds that we think may be harmful, and observe how the cells respond. This was actually how people discovered that you can take fibrillized alpha-synuclein, dump it onto some cells, and cause endogenous alpha-synuclein to phosphorylate and form Lewy bodies. With this method, we can also think about testing compounds that we think could prevent or reverse protein accumulation. So, for example, after Lewy bodies have accumulated, we can take a first step in looking to see if we can get them to disappear. In vitro experiments are popular for a few reasons, one of which being they are relatively cheap and easy to do. It doesn't cost a lot of money to have many dishes of cells in your lab. These experiments are also much, much quicker and can often be done in just a few days' time. This scale control also allows people to really dig into the nitty gritty of mechanisms, of the cellular mechanisms that we can't get into with full organisms. However, a really big caveat is that these experiments look at pieces of the whole. We aren't recreating full brains in these dishes, so we are inevitably missing parts. Parts that we know we're missing, and also parts that we don't even know that we're missing, which could actually be extremely important. This also means that the translational abilities, or the ability for us to take these experiments and translate them all the way directly into human bodies, is fairly weak. To bridge that gap from cell to body, people move into in vivo experiments or model systems. And we'll talk about a few different ways to model diseases, including genetic and inoculation models. Let's start small then with Drosophila, or those super annoying tiny flies that show up in your kitchen during the summer. These are a great small genetic model for diseases. And what I mean by that is that Drosophila in humans have 60% gene homology. So you and I are basically 60% fruit fly. But their small size is much more manageable for researchers if we actually compare chromosomes and base pairs on their DNA. This also includes a shorter life cycle. And there are many easy and quick phenotype measures we can test in flies, such as motor abilities or memories for different types of smell. In short, making things smaller also makes them much more manageable. However, small is also a limitation. This is a fruit fly brain, and if we take a look at a human brain and try to compare them, I'm going to say this is not even to scale. I could not literally get the fly brain small enough on the screen to accurately represent its real size for you to still be able to see it. So talking about pros and cons here, we just reviewed some of these, and another one that I want to point out is disease state. We don't see neurodegenerative diseases occurring naturally in flies, which means we have to use known genetic mutations in order to induce the disease, which takes away a little bit from our ability to study sporadic cases. Since size is a pro and a con, let's go a little bit bigger. Mice and humans have 92 to 97% genetic homology. So if you thought we were surprisingly close to flies, you know, in mice, genetic models are heavily used. And what do I mean by that? We have the technology to actually knock in human genes in a mouse, which we call transgenic mice, where we can literally find the human sequence of a gene we want, locate the mouse equivalent, and replace it. This is how we can study a risk factor for Alzheimer's disease, such as ApoE4. We can literally put it into the mouse. We are also able to insert known mutations that cause Alzheimer's disease, like in this mouse model that we call the 5XFAD mouse. This mouse has 5 of the known familial Alzheimer's disease mutation on the APP gene, thus the name 5XFAD, which means we can study the progression over time of pathology in this model system that is a full body, not just in a dish. You may have noticed one glaringly big limitations. Lily is very excited about all of this. But one of the big limitations that Lily really wants you all to know is that these are all genetic models. So, what about sporadic cases? These have been historically really tricky to figure out since we don't know why people get these diseases. This has recently been remedied, though, with inoculation models or what we call pathological seeding. You'll recognize this graphic from our neuropathology lecture, and I'm bringing it back because this is one way in which we can study sporadic Parkinson's disease. My mentor's lab, Dr. Uni's lab, has actually shown using these preformed alpha-synuclein fibrils, which we call PFFs, lead to Lewy body-like aggregation over time in mice. So this is a graphic from one of our previous papers showing the progression of time since we have injected PFFs into the mouse brain, down here, 2.5 to 4 months. At 2.5 months, we see this appearance of Lewy neurites, which over time mature into full-blown intracellular inclusions and continue to progress until these cells die months later. Now that we have created these diseases in models, we can start actually testing treatments in these models. And another pro to rodent research is that we have many behavioral tests that we can probe for cognition, for motor function, for social interaction, and many more types of behaviors that are altered in these diseases. Of course, though, these aren't perfect models. Similar to Drosophila, rodents don't naturally develop these diseases, meaning we're still not truly studying the disease itself. We are also limited to measuring only what we can observe in these animals, since we can't actually ask a mouse, "How are you doing?" Which are important things to consider when we're thinking about designing experiments. We're going to wrap up by briefly talking about non-human primate models. Since you got to do the virtual tour of the Primate Center we have here in Oregon, I really wanted to talk about these models in neurodegeneration research. Obviously, non-human primates are the closest animals to humans. We are primates after all. However, unlike models for neurodegenerative diseases in mice, there are no genetic models in primates, which means we have to focus on how we can model sporadic cases with seeding or toxin models. Now, I focused mostly on Alzheimer's and Parkinson's in this lesson, so I wanted to give a little bit more time to Huntington's disease, specifically because Dr. McBride is a researcher at the ONPRC who is working to develop treatments for Huntington's disease using her non-human primate research models. Now, Huntington's disease is genetic, but one difficulty is that we don't have the technology to create knock-in primates, like we can make knock-in mice. To address this, Dr. McBride has been using empty viruses to deliver this elongated mutant huntingtin gene into the primate's brains. So we can insert that mutant huntingtin gene into the virus and literally drop it into a primate brain. Once this is actually delivered into the brain and we can see the observed symptoms of Huntington's disease, we can start assessing novel therapies that may eventually lead to a cure for Huntington's disease. A much more common neurodegeneration model in primates is actually a toxin-induced model for Parkinsonism. In our previous lesson, I talked a little bit about pesticides and this chemical here, MPTP. MPTP was actually an accidental discovery. It's a variation of a chemical called MPPP, which is a laboratory made opioid agonist. However, if the chemist isn't careful, they can easily accidentally make MPTP. Several people who were known to struggle with opioid addiction in the 1980s suddenly appeared in the clinic in Northern California with immediate onset Parkinsonism. Remember, that's similar to Parkinson's disease, which was traced back to them having accidentally self injected MPTP instead of MPPP. Researchers immediately saw how this could be used to better study Parkinsonism and have used MPTP in primates to better understand what is happening, eventually leading us to understand the basal ganglia circuit to the extent we do now. Like with all of our methods, there are pros and cons though. Some I wanted to highlight include the fact that primates are much more advanced and can undergo more complex assessments that are closer to clinical assessments. On the other hand, we still can't communicate verbally with non human primates. And many people, including non human primate researchers themselves, think long and hard about the ethics of using non human primates. I can assure you that scientists care a lot about their research animals and go to great lengths to never cause undue pain or stress, and are extremely dedicated to never conducting unnecessary procedures or experiments. To wrap this up, there is no one perfect way to study neurodegeneration, and experiments in humans, cell cultures, flies, rodents, and primates have all led to very essential discoveries and will continue to do so.